Hey guys, welcome to today's notes. Last time we took notes, we were talking about some vocab that had to do with Mendel's genetics. And now we're going to talk about how to set up a Punnett Square. And most importantly, what is it that a Punnett Square can tell us? So it's easy to go through and complete Punnett Squares, but it's important to understand what it is that you're actually solving for. Otherwise, you're just doing work that you don't understand, and that's just a waste of time. So let's talk about first how to set up a Punnett Square and what it is that it's telling us. If you remember, we call this a karyotype. Let me make this a little bit bigger so you can see it. So here, remember that we have two first chromosomes, two second chromosomes, two third chromosomes. And on each, so since we have two first chromosomes, we're going to have a gene on each of those chromosomes. So let me draw out a gene here. I'm going to use this red color, and I'm going to draw right here. So let's just say that this is the gene for eye color, for brown eyes, or for blue eyes. So here, this is the chromosome that your mom gave you, and this is the chromosome that your dad gave you. And your mom gave you one version of that gene, let's say blue eyes, and your dad gave you another version of that gene or another allele. And let's say that's brown eyes. I forget what we said for your mom, if it's blue eyes or brown eyes. So let's say your mom gave you blue and your dad gave you brown eyes. So you have two versions of each gene. And what we do is we call that a genotype. So if mom gave this person, if this is this person, this is this person's karyotype, and their mom gave them blue eyes and their dad gave them brown eyes, well then their genotype for eye color is going to be big B, little b. And so here, we're taking this person, let's say that this is the, well, this would actually be a male because we have an X and a Y, so we'll put them up here. And we're going to put the wife or the female in this scenario on the other side. So we have two people, and they're going to produce an offspring. And what we're going to do is we're going to put the parents' genotypes on the outside. So this is this guy right here. We're going to put him up here. And we have the mom over here. And we're going to put her, let's give her a little dress. Really cliche. Here we go. And we're going to put her genotype on the outside as well. So the mom and the dad's genotype are on the outside of the Punnett Square. Let me go to the next page. Oh, it won't let me switch until I stop. Here we go. All right. So inside the Punnett Square here, this is where we're going to determine the probability of the offspring's phenotype. So here, it doesn't say that they're going to have 4 kids and one of them will have brown hair or brown eyes and one of them will have blue eyes. It's the probability. If they have one offspring, they have one child, what is the probability that their eyes will be blue? What is the probability that their eyes will be brown? That's what we're determining here in the middle of the Punnett Square, in the center of the Punnett Square. It is the probability of the offspring's phenotype. So we can see here that if that guy that I drew before and that girl that I drew before were to breed and have an offspring, there's a 75% chance that they would have a brown-eyed child and a 25% chance that they would have a blue-eyed child. That would be the phenotype. That is the offspring's phenotype. So remember, when we're talking about a genotype, we're using letters. And if we're talking about a phenotype, we're using a word. So here, the parent's genotype is big B, little b. the offspring's phenotype is brown eyes or blue eyes. So the phenotype is what we see. It's on the face, not necessarily, but that's how we can remember this phenotype face. And so the principle of dominance here is that if there is a capital letter, or if there's a trait that is dominant, we're going to mark that with a capital letter. So big B is, or brown eyes, is dominant over blue eyes. What do I mean by dominant? Well, if we have big B, well, those are both dominant. And so if big B represents brown eyes, then we have two big B's, two brown eyes, then it's definitely going to have, this offspring's definitely going to have brown eyes. But here we have big B, little b. So which one's going to be expressed? Is it the brown eye allele or the blue eye allele? Well, since brown eyes is dominant over blue eyes, then this dominant brown eye allele is going to dominate and be expressed over the blue. And so the blue right here is not the gene or the trait that's going to be expressed. It's going to be the brown. But down here we have little b, so blue eye, blue eye. There is no dominant allele. There is no capital B. And that's the only time that the recessive allele can be expressed, is if you have two recessive alleles, little b, little b. We call this the principle of dominance. And so the blonde hair trait is recessive, and therefore it can only be expressed if there is no dominant brown eye allele. And we saw that right here. For example, little b, little b. Now let's start putting some vocab to this. These are some big words, but big words are easy if you break them down into smaller pieces. So homozygous is the first word we're going to talk about, and that seems like a really weird word. But let's break this down. The prefix homo means same. So if we think about our Latin name, Homo sapiens, homo means same. That's us, same as us. So homozygous means that the two alleles are the same. Homo means same. So they can be homozygous recessive. This is homozygous recessive, little b. They're both the same. Or they could be homozygous dominant. So we have big B. They're both dominant. They're the same dominant. So if homo means same, then hetero means other. So the heterozygous is going to mean that we have both or other. They're different. So big B, little b. These alleles right here are heterozygous. Or if an offspring was born with big B, little b, they would be heterozygous. And we have this little comic strip here that shows that these sheep are heterozygotes, or these goats are heterozygotes. And they're both different, they're both two colors. And so that's just a little joke that doesn't actually exist. So those are the terms that we're going to use, and I want to go through a couple of questions on your assignment to help you through and to make it easier. So here we have our first sheet of our monohybrid cross worksheet, or our Punnett Square practice. These are going to be pretty self-explanatory, but let's go through here. Below each of the following words are choices. Circle the choices that are the examples of each of those words. So dominant allele. So here we're just going to circle everything that could represent a dominant allele, and that would be a capital D. Would this little e be dominant? Nope, that would be recessive. Little k, no. Big L, yes. So let's go down here. Genotypes in which dominant gene must show. Well, the dominant gene must always show. So we're going to circle everything that has a dominant gene or a dominant allele. So big A, yep, that's going to show. Big D, little d, yep, dominant allele. What about here, little f, little f? Should we circle that one? No, there's no dominant allele, so we can't circle that one. All right, so let's go to the next page. And if we were to set up a Punnett square, let me turn on my draw tool. Here we go. And we're going to draw our Punnett square. And in guinea pigs, short hair is dominant to long hair. So I like to write these out. This makes it easier. So big S is for short. And little s, so I'm going to make that really less, is for long hair. That helps me keep everything straight in my head. Complete the following Punnett squares according to the directions given. Then fill in the blanks. So one guinea pig is, so let's put some vocab here. Heterozygous. So big S, little s. So let's call that the dad guinea pig. So big S, little s. And the other one is homozygous recessive. Little s, little s. So let's cross this. We have a big S, little s, little s, little s. Big S, little s, little s, little s. So here, probability that the offspring will be homozygous for short hair. We have let me switch colors here, a 50% chance that, or 2 out of 4, chance that our guinea pig will be homozygous for I'm sorry, short hair is the see, it's a good thing I wrote it out here, is capital S. So we don't have any homozygous for short hair. So let's cross that out. Heterozygous for short hair will be right here, heterozygous. And so that's going to be 50%. And our long hair guinea pigs are going to be 50%. Or you could do 2 out of 4 or 2 out of 4. Let's go through. That should be explanatory. That should help you. Oh, let me turn off my draw tool. So that should help you with some of those. Let's go through, and I'm going to do number 17 for you, or with you, so that you can, because it's just a little bit confusing. There's a lot of steps. So in tomatoes, red fruit is dominant to yellow fruit. So a plant that is homozygous for red fruit. So let's draw this out. Red fruit is dominant. So let's go capital R is red. Little r is yellow. I'm just going to put yellow. So our P1, our parent generation, or our parent genotypes, a plant that is homozygous for red fruit. So homozygous for red, so big R, big R. And we're going to put an X here, and this means that they're going to breed, is crossed with a plant that has yellow fruit. So if it has yellow fruit, it's going to have at least one little r. But if it had a big R, then the big R would be expressed and it would be red. So in order for that recessive trait to be expressed, it's going to have to be 2 little r's. And so the phenotypes, so those are the 2 genotypes, and the phenotypes are red and yellow. So what will be the genotypes and probabilities of the phenotypes of the daughter generation? So let's do this out. If we have big R, little r, little r, we're going to have, I don't have enough room here, big R, little r. Let me make my Punnett square much bigger. So big R, little r, big R, little r, big R, little r, big R, little r. So we have a 100% chance that so the generation genotypes are going to be big R, little r. All of them are big R, little r. And they're all going to be, since they're capital R, they're all going to be red, so 100%. And no yellow. So this is our F1 generation. 100% of our F1 generation is going to be red with the genotypes big R, little r. Now, if we took 2 of these tomato plants and crossed them, so if we make another Punnett square, big R, little r, we're going to make this, big R, little r. Then we have big R, big R, big R, little r, big R, little r, little r, little r. This is our F2 generation. And so here our F2 generation is going to be, what are the genotypes? I would write out big R, comma, big R, comma, little r, little r. And for our F2 phenotypes, we would say that they are 75% red and 25% yellow. So that is going to be how much did I help you on this. So finish these, take a picture of it, and upload it to PowerSchool. I hope this helps. Let me know if you have any questions.